Infectious Diseases Tock Seng, and Applied Genomics Lead in NCID Singapore. He also holds a joint academic appointment at Lee Kong Chian School of Medicine, and his research interest is obviously genomics for infectious diseases and antimicrobial resistance. And thanks, Oon Tek, for agreeing to give this talk. And with us today, we have two panelists, and they're both exceptional in their field. Prof Anucha obviously needs no introduction, and Prof Anucha is the Professor and Chief of Infectious Diseases at Somerset University Hospital. He also serves as an adjunct visiting professor at Division of Infectious Diseases, Washington University School of Medicine, USA. And Anucha's research focus includes IPC in resource-limited setting. In fact, he does many things. What I'm saying is probably just a fraction of what he does. He also sits in various national and international committees and guideline groups, and also editorial boards of journals, including ICHI and American Journal of Infection Control. And thanks, Anucha. And all the way down in south, we have Dr. Max Bloomfield. Dr. Bloomfield is a consultant microbiologist and infectious disease physician at Wellington Southern Community Laboratories and Capital Coast and Hutt Valley, New Zealand. He has recently established a genomic surveillance program for local IPC using Oxford Nanopore sequencing, and his other research interests include diagnostic stewardship and using large epi data sets to inform and improve clinical practice. So thanks, Max, for making time and joining us today. So I think it's going to be a very interesting talk and panel discussion. So Oon Tek, thanks. The Zoom is yours to start your talk. Thank you. Okay,
thanks a lot, Kailesh, and it's a pleasure to be able to share on this topic, whole genome sequencing for IPC. I'll limit the talk to 30 minutes, and also a pleasure to share the panel with Dr. Prof Anucha and Max as well. So today the topic is whole genome sequencing for IPC. I'll try and cover what its advantages are, touch a bit about its disadvantages through some examples, and also a bit on the cost effectiveness.
Sorry, Oon Tek, just one thing. Participants, please put your questions in the Q&A section. As you have them, I'll collate them. Thank you.
Okay. So, yeah, do stop me if the audio or anything is not working well. We'll cover these broad sections. So I'll proceed to just give an introduction on genomic linkage. For those who are familiar with this, please bear with us. For those who are not, I hope that it's informative. So very simplistically, a bacteria can be thought of as having a chromosome that's in a nucleoid region. And also the green circles represent plasmids in some of these bacteria, especially gram-negatives, but also present in gram-positives. Now the chromosome as well as plasmid can be thought of in one sense as the alphabet soup or alphabet line, and essentially can be summarized in 4 of the nucleic acids. And approximate range of the length of this for the chromosome is from 2 to 5 million of these ACTs or Gs, these alphabets. And the plasmids as well, although smaller, usually 50 to 250,000 bases. Now, in whole genome sequencing, one of the ways we can think about it is that we are trying to determine the genetic alphabet. You can see that to determine 5 million alphabets at one go is not an easy task, but the technology has improved a lot. And so now it's possible. For infection prevention, for linkage, usually one of the central concepts is that of the single nucleotide polymorphism, or SNP, essentially a change at a single position in the sequence. So if we could just represent it in this way, for example, in this particular genome comparing 2 bacteria, if there's only one difference in their genetic code, let's say over 5 million bases, a C becoming a T, we'll call that one SNP. And usually for determining whether they are descendants of each other, and therefore likely to have clonal transmission, the SNP count has to be low. Whereas if there are more SNPs, let's say the middle and the lower example, there are 5 changes, roughly, we can call them 5 SNPs. When the SNP count goes higher and higher, it suggests that the bacteria are not linked. And usually in most contexts, the threshold ranges approximately 10 to 20 SNPs in the time frame of study. Now, in whole genome sequencing, what happens is that the bacterial genome is first digested into small fragments because it's very difficult to read the 5 million bases at one go. That's why whole genome sequencing is also called massively parallel sequencing, because there are millions and millions of these small fragments, which are then loaded into either usually Illumina nowadays for short read and Nanopore for long read, but there are other companies as well. We're not partial to any of them, using them just as examples. And what these machines do is that they sequence these many small random fragments at the same time. And that's why they manage to sequence millions and billions of bases over a few hours or a day or two. Now, a large challenge of the bioinformatics, which there are a lot of tools that have been developed, is to actually piece these fragments back together to make a meaningful genetic code. Okay, and in this case, for example, if you take the red line, if you can see my pointer, may correlate to the red sequence, the green one here. So basically, due to related to similarity of the sequences, we can actually what we call assemble these sequences into a long contact. And from there, once we've got a semi-whole or whole genome, we can then use computational tools to determine the SNPs, linkage, call AMR genes, and so forth. Now, if we've dealt with the concepts, a quick question is, what does whole genome do in the real world for IPC? Does it work? There are many, many examples, but I chose just to highlight one more high-profile example that was about 10 years ago, where the US NIH Clinical Center had an outbreak initially over the first 6 months of 18 patients with KPC positive K pneumonia, of whom 11 died. This was subsequently published and also reported in the popular press. Now, without whole genome sequencing, the point of this figure is to show that the linkages are relatively difficult to discern. It looks quite messy in this figure C, where there are many, many potential linkages, and without any molecular tool or lab tool to link them, we usually have to determine based on where the patients have stayed and whether they have potential spatial overlaps with time. And that's why the linkage map is quite difficult to discern. But integrating whole genome sequencing, you can see that those complex linkages then are simplified because based on the SNP counts, they'll become apparent usually when you combine it with the epidemiologic information, where the most likely linkages of transmission are. Now, the bad part is that just to note, whole genome does not replace conventional epidemiology information. It must be combined with the usual spatial-temporal and other information about the host in order to be meaningful. We will then go on to share two particular examples that we have embarked on in Singapore to illustrate this concept further. Now, the first example is whole genome in a retrospective, relatively large population setting. And this was performed to determine transmission of carbapenemase producing Enterobacterialis. In Singapore, as in many parts of the world, CPE numbers were increasing. Still, I think now are gradually increasing. Some parts may be plateauing. And in Singapore, the main variants that we have, the main genes would be KPC and NBN with a smaller proportion of OXA. In terms of species, as in most cohorts, with Klebsiella pneumoniae as the predominant one, followed closely by E. coli and then Enterobacter. Now, in order to further understand the genomic correlation, we just have to introduce the concept of horizontal gene transfer via plasmids. And this is where genetic determinants, including the carbapenemase gene, can be spread from one bacteria to another, not by progeny or descendants, but by actually gene sharing through a mechanism called conjugation. Now, the conventional way of spread that we think of is usually clonal transmission, where if you look at a person in red, he has the bacteria that carries the offending gene. So in this case, the CP gene carrying bacteria, and the bacteria can spread directly to a new person, the person in black. Now we know that this clonal transmission can also happen via intermediate reservoirs. For example, in this case, we illustrated it using a sink, where the bacteria could have moved and his progeny moved from the person in red to the sink, and then from there to the person in black. Now, in plasmid-mediated transmission, it is different in that the gene actually moves from one bacteria to another bacteria. So in this case, the blue color one to about the orange color one, and subsequently the orange bacteria then moves to another host. And of course, in the top diagram, we have simplified it. This spread can also happen via intermediates, and it can get relatively complicated. Now, one of the nuances to highlight about plasmid-mediated transmission is that it can become apparent that if we use the SNP approach to link two bugs, we would miss plasmid-mediated transmission because this bug, the blue one on the right-hand side, is clearly different from the rectangular one on the new host. But however, they do share transmission via horizontal gene transfer via plasmid. The other nuance for those who are familiar with this is that usually plasmid determination, at least of this stage, requires a bit more work to fully close the genome and not just have a draft genome. So in the case of our project, we wanted to determine why CPE transmission was increasing in our hospitals, even though infection prevention measures were being implemented. Specific questions were, is there significant prevalence of clonal and plasmid-mediated transmission, especially plasmid-mediated transmission? Number two, are there epidemiologic differences comparing these two genomic modes of transmission? And number three, are there differences in response to population level IPC for these two genomic modes of transmission? Now, our study was conducted over 5 years. Thankfully, due to work of Kalish and other colleagues in the CAPES network, it had very relatively good coverage. Most of the government hospitals were on board, in fact, all multidisciplinary hospitals. And there was mandatory referral to a central collection point at that stage. So this covered 80% of inpatient care in Singapore with about 8.4 million bed days at that time. Very quickly, they were sequenced both on Illumina and Oxford Nanopore for long read. And the similarity for clonal transmission was using conventional SNP criteria. For plasmid-mediated transmission, we had to call the plasmids and then determine plasmid clusters. This is a bit of a disease slide, but essentially we had 1,300 isolates from 780 acquisition patients. Finally, about half of those that could be genomically assigned were clonal transmission, and the other half are plasmid-mediated transmission. And these are just the clinical and microbiologic characteristics. As we expect, they are older patients. Close to 70 years old was the median age. And length of stay was about 5 days for median. About 2/3 were surveillance cultures and 1/3 clinical cultures. Now, first comparing clonal transmission and plasmid-mediated transmission clusters, we can see that clonal clusters tend to be more in the population, but with less patients per cluster, and they persist for shorter time. And they involve less hospitals compared to plasmid-mediated clusters. We then went to determine the contact types that were associated with the different genomic modes of transmission. So in the first instance, clonal transmission. Now, just to remind the audience, this is a case where the same bacteria can be demonstrated via whole genome to have moved, whether directly or through intermediates to another patient. We look for 4 types of contacts. So ward and hospital contact, procedures, mainly scopes, endoscopes, and uroscopes. Discipline, where the person shared a medical or surgical discipline, taking care, providing the clinical care, and then community contact. So for clonal transmission, what we determined was that the spatial-temporal contact was actually important. Direct ward contact, where the 2 individuals shared the same ward location at the same time, was the strongest association. But interestingly, we also showed that even if they were not in the same ward or even the same hospital at the same time, there was an association suggesting that this bacteria could transmit through hidden reservoirs, whether intermediate patients or the inanimate environment. Reassuringly, at a population level, we did not find procedure to be a significant risk factor, neither did we find discipline or community transmission to be likely significant. Now, the picture was slightly different for plasmid-mediated transmission. Just to remind everyone, in this case is where the linkage is via the extra chromosomal DNA, the fragment, usually 50 to 250,000 bases, and it can move horizontally between different bacteria. For simplicity, in this illustration, we just showed 2 patients, but again, this could have happened through many intermediates, including the inanimate environment. Now, in this case, the epidemiologic association using adjusted odds ratio showed no significant contact to be statistically significant in transmission. This suggests that the plasmid is widespread, and we cannot determine a direct epidemiologic correlation from one patient to the other. We then went to examine whether there was difference in time trends. Now, in this graph, we see plasmid transmission. Bacteria represents the clonal transmission, and unlinked, thankfully a minority, about 10%, is where we could not assign it to plasmid or bacterial transmission. Now, in the graph, we can see also a background gray area, and that was the number of surveillance cultures or density of surveillance cultures that were performed. And this served as a surrogate of the intensity of IPC measures as well against CPE. Now we can see that over time, both plasmid and bacterial-mediated transmission increased. However, towards the second half of the study, bacteria-mediated transmission actually decreased, and this was significant. However, plasmid-mediated transmission showed no evidence of decrease in the study. When we stratified it to surveillance and clinical cultures, we see that the effect is mainly driven by the surveillance cultures because it forms the bulk of the numbers. We then went to stratify via ward and hospital modes of contact. Direct contact representing that 2 patients were in the same ward at the same time. Indirect contact meaning they were same ward but at different times. And with hospital direct contact meaning being in the same hospital at the same time, and indirect contact meaning being in the same hospital but at different times. And we found that the decrease in clonal transmission could be mainly explained by a decrease in direct ward transmission, suggesting that the infection prevention measures were mainly effective in the direct ward level. We did not find any decrease in plasmid-mediated transmission in any contact type, mirroring the fact that we did not see any decrease in plasmid-mediated transmission at all. So what does this all mean? Well, firstly, we can confidently say that plasmid-mediated transmission is significant at a population level and has to be accounted for if we want to obtain a complete understanding of CPE transmission. We can also say that there are clear epidemiologic differences between plasmid clusters and clonal clusters, in that plasmid clusters, mediated clusters appear to persist longer, involve more patients, and involve more hospitals together. Now, in ecologic correlation with population level IPC measures, we can suggest that the decline in clonal transmission is mainly due to direct ward contact related transmission, and IPC measures potentially are most effective there. Now, this is a graph showing the different measures in the different hospitals. And we can see that all hospitals had terminal cleaning, ASP programs, hand hygiene, contact precautions, and single room isolation. Now, for more, I would say, higher level or specialized type measures like regular surveys, high-risk screenings, or hydrogen peroxide vapor cleaning, there was variance in enactment across the different institutions. Now, further implications for the transmission of CPE, our data supports the literature suggesting that the hospital environment is a reservoir for CPE, and that a person could get it from the environment without having to get it from another patient directly. As we mentioned, for those that are using whole genome to determine CPE transmission, we suggest that plasmid linkage also has to be accounted for, especially at a population level. So we then move on to our next application, where we use whole genome for actionable results in the context of carbapenem-resistant Acinetobacter baumannii. Now this is published in IPC, and essentially what happened was that in 2016 we noted an increase from baseline in the hospital, and an effort was made to sequence environmental and clinical samples over a 6-month time period and combine it with the previous data that we have to understand transmission and find where the reservoirs are for intervention. In summary, the sequence data after analysis determined 5 clonal clusters. I'll focus on the one with the highest number of isolates and patients. The rest of the clusters also had the same findings. Now, after determining transmissions using the SNP count, we then went to the spatial-temporal overlap data to determine where the most likely place of transmission was. And you can see that for patients with obvious spatial-temporal overlap, the most likely place of transmission was actually the MICU or the medical ICU. This then resulted in sharing of the data with the IPC unit. In the first meeting, the whole genome data was shared, and it was then agreed that epidemiologic analysis and correlation and further whole genome sequencing was to be done, and this was performed. Subsequently, about 3-4 months later, it was then became more certain based on added data that MICU was the main focus of horizontal transmission, and enhanced IPC interventions should be instituted. It was then decided that the findings should also be shared with the hospital ICU committee to determine the best approach to intervene. And this was performed. This sharing was done about 3 to 4 months later. And enhanced terminal cleaning and decontamination was one of the measures that was suggested. And this was then enforced and performed. Now we can see from this ecologic graphs that the presentation to ICR IPC committees happened with the middle of 2017. And it was then agreed that hydrogen peroxide vapor should be increased, the use of it in the hospital, and this actually did happen. And it was also associated with a decrease in positive CRAB clinical cultures across the hospital, as we can see on the bottom graph, thereby providing some ecologic support that the enhanced measures actually made a difference in the numbers and in transmission. So after having shown that whole genome does add data to IPC and can be used for action, although it does take a bit of time, one of the other questions we are commonly asked is this cost effective? Is it too expensive? Now, there are not too many studies, but I selected studies from different continents, and across Australia, US, and Germany, in the limited data we have so far, it does suggest that it is cost effective, especially in resource enabled settings. Now, roughly, these are ballpark estimates because we are also commonly asked, what is the cost per isolate? It can vary quite a lot depending on the circumstance. However, I would suggest ballpark right now. Per isolate sequencing costs for just short read closure would range from $50 to $100 US. Most settings may charge that same amount for computational analysis. So with that, I'll close this talk and just summarize. We introduced the concepts of whole genome sequencing and its utility. Does it work in real world settings? I would suggest yes. There's a lot of data and publications, but I think the time doesn't allow us to do a systematic review. We also shared our own experience in Singapore with CPE in a population. We shared a single hospital experience with interventions related to the whole genome data and epidemiologic correlates for CRAB. And in terms of cost effectiveness, yes, the data at this stage at least suggests that in at least well-resourced settings, it is cost effective. So with that, I think it just leaves me to thank the audience for listening, as well as especially all the collaborators in the CAPES network, predominantly infectious disease physicians and microbiologists, maybe some of whom are in the audience. Thanks for all the work on these projects. And I'll hand the time back to Dr. Kalishwa.
All right, great. Thank you so much, Oon Tek. That was really excellent, and I mean, I learned many new things as well. So we'll go to a Q&A session. All right, great. We are almost at the end of the session. Any last comments? Comments from the panelists? No. Okay, great. Then thank you so much. I think this has been a very interesting session. I thought it would be boring because I've heard it so many times, but I think this has been very interesting because the comments that came, especially they are very good. And thanks Oon Tek and Max and Anucha for participating. And yes, I think this has been very useful. And just to everyone's information, there won't be any IPC club session in December. It's a holiday month. Season's greeting to everyone, and Happy New Year. In January, we will have, I think Andrew Stewart is online. So Andrew and I will be chairing a panel session in January where we will actually invite maybe 8 to 10 IPCs from Singapore and the region to give short snippets on what they think is important change to IPC or IPC should look forward to in the next 3 to 5 years. I think it'll be interesting. So we will keep you posted. Until then, thank you so much.